Tēnā tātou koutua, ko Grant Smith, tōku inora, no Otutahi aho. I'm a plant pathologist based at Plant Food Research in Lincoln. Today I'd like to talk broadly around our work to understand how this pathogen causes disease and what we're doing to now try and target some of those putative pathogenicity determinants in the pathogen. Whoops, why won't? There we go. So this work actually basically started in 2017 in terms of actually being able to understand resistance, that we have resistance in plants in New Zealand, and also we needed a genome to do the type of work that we've been undertaking for the last few years. So the beyond myrtle rust transcript studies will be the focus, but there's some stuff before and after. And I'll also touch briefly on the work that we're doing that's funded by Biological Heritage National Science Challenge around exploring double-stranded RNA interference as a putative control measure for myrtle rust. So most people will be familiar with our long-term colleague Louise here. She is inoculating plants, New Zealand plants in facilities at Putative in Brisbane. It's about 2019, I think this photo was taken. It was this material which gave us the basis for describing, but not only whether we did have resistance, but also the material that we used to then explore the molecular basis of that resistance to myrtle rust in New Zealand taonga. Now, one thing I would like to address as part of this is there seems to be this ongoing misperception that Mānuka is resistant to myrtle rust. That is not the case. In our work that we undertook, basically 85% of the plants that we tested are susceptible to infection by the pathogen. The fact that we don't see it on the landscape is probably more something to do with the epidemiology of the disease rather than the Mānuka having some broad level resistance. What we did find that there are two types of tissue-specific resistances in Mānuka. One's leaf and one's stem. And more recent work by David Charno that he's presented showed that they are localizing to two different chromosomes in a SNP analysis work. The two leaf resistances are the immune or the hypersensitive, so what we call L1 or L2, and the stem resistance appears phenotypically at least to be an immune type response. So 75% of the families that we tested had resistance, either L1 or L2, but only about a quarter of those plants from each of those families had those resistances. So most of the plants, even in families where there is resistance, are not resistant. And some families only produced either L1 or L2. But at the end of the day, 85% of Mānuka plants are susceptible to leaf or stem infection. And it's similar for Canuka, that around only about 15% of the plants that we tested are resistant. Whoops, done it again. Okay, so effectors are relatively small and cysteine-rich proteins. They're generally associated with the manipulation by the pathogen of the host. So they suppress plant immune perception. So they suppress the plant's ability to know it's being infected, and then they alter the host physiology for pathogen benefit. So those nutrients that are produced by the plant cell are exported out for the pathogen to grow on. So recently the genome was phased. So it's now di-haploid, and the current estimate is that there are over 1,200 candidate affected genes in the Austropuccinia genome. And the differential gene expression, so differential expression, it means it changes, the amount of expression changes over time. They tend to run in curves or pulses. And this is a very generic model of gene expression in rust. And you can see those effectors and other secreted proteins in the orange tend to come as a series of pulses throughout the infection process. So the work we did was try and understand that, and basically at first experiment where we took samples at 24 and 48 hours is the two main drivers of, so the analysis says that there are two main clustering factors. One is time and the other is family. And so you have to realize that we're dealing with uncharacterized germplasm. So the families are families are noted throughout the process, but basically, so the families are responding with the genetics they have to the infection, and that and the gene expression of the plants changes over time as they resist or are susceptible to infection by the pathogen. When we look at what the pathogen's doing, the pathogen, the main principal component analysis says that basically time is the key driver. Families are less important, and the samples tend to cluster by time. And so it's a wee bit like one of those stages in terms of that differential expression model that for us, in terms of saying that the expression of genes has changed over time, which is important in saying that you've moved from a first phase into a second, or a second phase, at least in this experiment. What we were able to do was identify at least 3 putative effectors, a cellulase type gene, a mycotoxin production enzyme, and a cell wall enzyme as being the top 6 differentially expressed genes in that 24 to 48 hour phase of infection by germinating spores. And a more detailed analysis, and this is from the first experiment, 24 hours to 48 hours, is that there were 16 putative effectors that had significant differential expression patterns, which means that the amount of expression changed significantly over that time period. The phenotype of the host plant is not important, and that's important because it suggests that the pathogen is not reacting to the plant. It's not changing gene expression, whether it's on a resistant plant or a susceptible plant, which suggests at least that the expression of the fungal genes is predetermined, and it's following a very set process in terms of infection, in terms of timing of expression of those genes. Now, a couple of genes here, 5292 and 1260, are being investigated as proteins in their own right by students at the University of Canterbury. So this is Nikki's work to understand 5292. This one's quite an interesting one in terms of the number of cysteine residues that are present in it. And Nikki presented this work at the Sydney Myrtle Rust Conference, and that is and currently these, even though they're relatively small proteins, they are proving quite difficult to actually analyze as proteins. The second one, 1260, seen that Yavan is investigating and presented on in Sydney. It seems that it's localizing to the endoplasmic reticulum in the plant cell, but we're still trying to get a sufficiently robust model of how that protein is working at this point in time. So we've moved on from our first set of experiments and had a look at transcripts being expressed a lot earlier on, before 24 hours. And in 1260, we can see that the effector is expressed a lot earlier than 24 hours in the germinating spore, and we see an increase in transcript level, and particularly in the L5s, which are the very susceptible plants. We don't see so much of an increase in L1 or L2 because remember these are actually resistant phenotypes, and so the spore is actually germinating. It's using its own resources. It's not able to draw on resources from the plant because it can't establish an infection. All these data tend to be a bit noisy because we're not dealing with true biological replicates. We're dealing with families, subfamilies, plants and subfamilies, which, you know, we know the genetics of, we know where the seed came from the mother, so we know they share 50% of their genetics from the mother, but we don't know where the pollen actually comes from. So we do get these fairly noisy type of graphs. Second effector, what we call 143. This is an experiment where we took samples 20 minutes after inoculation and you could see sort of reasonable levels of effector, which almost suggests that this effector might be this transcript might be preformed for this effector, which suggests perhaps it's got something to do in terms of being a very early important protein. Because transcripts have to be translated into proteins in the infection process. So about 28, 29 months ago, the Environmental Protection Agency redetermined that double-stranded RNA, or RNA interference as sometimes known, is not a genetic engineering technology as defined by the Act in New Zealand, which meant that we had an experimental system that we could actually look to test some of our hypotheses around some of these transcripts by interfering with the production of RNA by the pathogen. So RNA interference, it's usually explained by a defense-based hypothesis as a defense mechanism against transposable elements in RNA viruses, which attempt to invade eukaryotic genomes, and it was present in the last common eukaryotic ancestor. So it's a very old and ancient mechanism for defense against virus infection of genomes. Now, various forms of RNA, including double-stranded RNA, can be used to induce RNA interference, and this technology's got fairly wide applications, including in human medicine to treat basically what is an autoimmune. It's a hereditary condition in human adults. In agriculture, there are two main forms of RNA interference. This one's called SIGS, spray-induced gene silencing, and the other one is HIGS, host-induced gene silencing. HIGS is a transgenic technology. It needs the gene to be introduced into the host, whereas SIGS can be applied topically. So most audience will be familiar with presentations by both Beck and Anne around the early work done in Brisbane around targeting housekeeping genes of Austropuccinia, and the success that was demonstrated by double-stranded RNA SIGs for myrtle rust control. Basically, targeting in particular the beta tubulin gene results in a significant decrease in the ability of the pathogen to infect the host plants. Now, all of these technologies have some variants in them, and you can see in that first A graph that extinction factor 1 and 28S, which is part of the ribosome complex, targeting those had more or less control than targeting beta tubulin. And the GFP is basically a negative control to show that just introducing any double-stranded RNA isn't going to introduce that effect in the plants. So with our Australian colleagues, so these initial experiments were actually done in Brisbane. We investigated whether we could target pathogen effectors in the attached leaf assay that they were using in Brisbane. The basic answer is yes, we can, and targeting this particular effector, which in this slide is number 3, gives significant control over, prevents infection by Austropuccinia. You can see a little bit of noise in that leaf replicate 6, but basically this one looks like a very interesting effector, and basically it poses the question, is that an effector? Is that effector a key effector in the establishment of the biotrophic interaction between the plant and the fungus? You can also, the assay also ran an in vitro spore germination assay, looking at targeting how it affected spore germination by targeting that particular effectors. So there's another couple of effectors in there. And you can see a reduced spore germination when you target the germinating spores with the double-stranded RNA, which does raise the question, even though these are classified as putative effectors, i.e. they are important in establishing the pathology of disease, establishing disease, do they actually have other roles in the biology of the pathogen? And at this point in time, that's a question that remains unanswered. So we've now got everything up and running now in Lincoln, and are now running detached leaf assays at our containment facility at Lincoln, using Cisginium, which is the same plant that our colleagues in Brisbane use as the test plant for exploring all the effector opportunities that we have. Particularly like to note colleagues Shay, Jen and Rebecca for all the work they've done to get us up and running in our facility at Lincoln with this technology. So in summary, fungal transcription appears to be independent of host plant phenotypes, so that's really important because it means it suggests at least the fungus is not changing its gene expression because it's on a resistant or a susceptible plant. And so any targeting that we do should be broadly applicable for disease control. Transcripts for some putative effectors may already be present in the ungerminated spores. Still like to do some more work around that, but it does suggest that those encoded proteins are absolutely essential for initial or first stages of infection, because they're basically ready to go. There are significant challenges working with these non-clonal plants. We get quite noisy data. We can see differences between the families, which we would expect. But we do not know the status of the SIB relationship. So the plants that come from single family, we do not know where half the genetics come from. We're hoping they come from the same pollen donor, but we don't know. So double-stranded targeting of one putative effector significantly reduces symptom development. So it does beg the question: is this a key putative effector in the establishment of the biotrophic interaction? And targeting 3 different putative effectors affects spore germination, so that we do know that they could be having other biological effects. But what this work is demonstrating is we can target not only housekeeping genes, but also what we consider to be pathogenicity determinants for putative control of this pathogen. And with that, I'll conclude, and I can answer any questions. Thank you.
Thanks, Grant. That was really, really awesome. Audience, if you have any questions, please put them into the chat box. There are some compliments coming in, flowing in for you, Grant. What an awesome talk and nice results. So I had a couple of questions for you. So you mentioned at the start that mānuka, the resistance and susceptibility was different from mānuka. How was it different?
Oh, so Canuka's, there's a third infection phenotype on Canuka. So on Mānuka, it's leaf and stem. On Canuka, it's leaf and stem, and also there's a tip dieback symptom that's also seen in some of the. So clearly the interaction of the pathogen with those 2 hosts in particular are throwing up different, or they're throwing up different phenotypes, and then we know, at least with Mānuka, that from David's work, that there's probably at least 2 independent genes that are associated with those two different resistant phenotypes. So Mānuka is challenging, Canuka is almost impossible in that respect.
I was good, yeah, because my question after that was that is that like the next steps is to try some of these other species?
Well, right now we're not working with Mānuka. We're only working with Cisgenium right now. We will need agreement, discussions and agreement before we actively go and work with taonga species. Yep.
All right, so a question is, have you tried double application? Is it possible to target two different effectors at the same time?
Not yet. There's a long list of things we haven't done. But yes, I mean, I think we'd also like to try and see can we combine what happens if we combine one of our effectors with one of the housekeeping? Are we going to, especially one of the ones that that doesn't give complete control? Will it pull it down? Will it help? And is it a mechanism to step around putative evolution of resistance to targeting these particular gene sequences? We don't know yet.
Sounds really interesting. Another compliment. Excellent work. So exciting to see these early expressed putative effectors in the RNA sequence, in the RNA-seq. So is in terms of the genotype, because you mentioned that the phenotype isn't important, but is the genotype of the host plant important? Are you expecting to see differences if you use in different species?
No, I don't think so, because we all of this data that both we and our Australian colleagues have got has been off Cisgenium, largely because it's such a good plant. So it is easily infected, but it recovers reasonably well. So it's a good experimental plant for us, but at this point in time, we certainly haven't tried investigating any other plants. We've got to get the system up and running properly with Cisgenium, and again, like I said before, we need to have lots of discussions before we can start working with Mānuka or any other taonga species. Yeah.
All right, another question. Nice work, Grant. Have you considered using DNA to investigate relatedness among individuals within and between host families?
Well, this was, David did some of this work and presented it a while ago in terms of using SNPs to try and analyze some of the diversity and within a family. But that data is still pretty preliminary, and and the main driver for that work was to try and see whether we could determine that there were two separate loci for the two different phenotypes, at least in Mānuka. So yes, it's important, but right now trying to deal with we're dealing with an undomesticated host, so we do not have any breeding history. We do not have a long history of knowing what the pedigrees of any of these plants actually are. So it's a big challenge, but it hasn't stopped us just trying to crack on and just have a go. Yeah.
Yeah, no, and I think a lot of people are rooting for the fact that this RNAi seems to be a really awesome novel way to control the disease. So in terms of the RNAi work, what are the next steps?
Well, I think one of the things we need to be slightly cautious of is that the current method that we have of applying double-stranded RNA is SIGs, which is a topical application, and the working hypothesis is it's actually targeting the fungal genes. The RNA mechanism is being self-processed by the fungus. It's not a plant-driven process. That's our hypothesis. What we don't understand is how often we might have to apply that treatment, because it's a topical. It's an application to the outside of the plant. most of the literature doesn't talk about those molecules being able to travel into the plant, and it's probably less likely with something like Myrtaceae, where you've got a lot of waxiness and sometimes oils and things on the surface. So we've still got a lot of understanding about what's actually happening, how long that double-stranded RNA will last. Double-stranded RNA is a relatively tough molecule compared with single-stranded RNA, but it will degrade, especially with UV in the environment. And so part of that is how you actually apply it, particularly if you're going to attempt to apply it in a very mixed foliage type of situation. So this is not we're not doing experiments on wheat that we know was this high and it's all genetically uniform. We're doing experiments on plants where we do not know what their genetic lineages are. Yeah. There's lots of challenges for Marsh.
Don't I know it? Funding is one of them, I think. We had much lower susceptible rates from controlled inoculation of Leptospermum scoparium from south to north. It's morning. Anyway, in Australia, do you have any thoughts on that?
Sorry, what was it? Didn't quite catch what the question was.
Sorry, with me laughing. We had much lower susceptible rates from control inoculation of Leptospermum scoparium from southern New South Wales. Do you have any thoughts on that?
Well, the latest understanding I have of Leptospermum scoparium in Australia is it's probably actually a different species from. And I believe, if I understand correctly, it was named in New Zealand first, so that means the Australians will have to change the name of whatever they got. So it's a different. I know that there's always been a lot of controversy about the same name for plants which are phenotypically quite different, but I understand now that there's actually reasonable evidence to suggest that they are actually different species. So it's a different species, so it's going to be different. I guess is the easiest answer for that right now.
Yeah, sounds good. Well, I think that is it for today. Thanks, Grant, for your presentation. So video of today's webinar will be made available on the Beyond Myrtle Rust website in the next couple of days. It's also going to be emailed to everyone who has registered. Our next webinar is on November 15th. At 11, as usual. We'll hear from Albie Marsh, who's the Maori Relationship Manager at Plant and Food Research and leader of the Kaitiakitanga and Maori-led solutions at Beyond Myrtle Rust. So keep a lookout for that webinar invite next month. Thanks, everyone. Hai reira. See you next time.